So hello everyone, welcome back to Hasvora Education. Today we are going to be solving the expected paper 2025 P34. In this paper I had just compiled two questions, one from the microscopy section and one from the dilutions, in which we are going to be performing an experiment based on an enzyme. So now let's begin. The first question that says that the progress of some enzyme catalase reactions can be followed by measuring the time taken for the substrate to be hydrolyzed. The enzyme amylase catalyzes the hydrolysis of a starch to maltose. So now let's just underline the important points. You will investigate the effect of different concentration of amylase on the hydrolysis of starch. You are provided with the material shown in table 1.1. So let me just adjust my pen first. So they have shown us the solution that we are being provided. The E is 2% of the amylase solution. The hazard that they have shown us is irritant. So we know that risk involved with the irritants I have already showed you in the video of the revision, so that the irritants have a medium risk. The S solution, 1% starch solution, none hazard, and I have also provided the name together with the risk in that video, that the starch solution has a low risk. After that comes the water, the iodine. The iodine has a medium risk as it is also an irritant. So watch that video if you do not know the risk involved with the solutions. So as they usually come up in the paper. So if any solution comes in contact with your skin, wash off immediately under cold water. It is recommended that you wear suitable eye protection. Think about the hazards of using the materials in 1.1 and decide whether the risk of using ES and iodine is low, medium, or high risk. So we have already completed the table. Then they are asking that you will need to prepare different concentration of amylase solution. Record the time taken for the amylase to hydrolyze starch. Then you will use the result to estimate the concentration of unknown solution of amylase U. So we are going to be doing in this in the end. You will need to use proportional dilution. So many of people are actually just confused in the proportional dilution as they have never heard of it. So proportional dilution is just more of like, uh, you can say, similar to the simple dilution, as we just decide the concentration that we are going to be making. So to make different concentration of the amylase solution E, you will need to prepare 10 centimeter of each concentration using E and W. So W is the water and E is the stock solution, or you can say the original solution. So table 1.2 shows how to prepare the two of the concentration you will use. Decide which other concentration amylase solution you will use. So now they have provided us with the table. Now you can see that the percentage concentration of the amylase solution that in the starch, where in the start, which is the stock solution, or you can say the original one which they have provided us. So we will be you can see the last one, the last most concentration that they have just given us is 0.4. So we can just see that what gap are they actually trying to convey to us, that what gap should we uh, just uh, keep between our concentrations. So as we know that we are going to be making four concentrations, so we can divide and we can multiply zero point 0.4 into 4, which comes out to be zero point 0.6. So it means that this one is going to be zero point 0.6. Then after that the other one is going to be, let me just write it a bit below, so it will be 1.6. This will come as 1.2, and the other one will 0.8. So we know that the gap will be about 0.4%. So this is how we actually deduce that how we are going to be keeping the gap between our concentration. So now they have provided us with the volume of E, that is the 10 centimeter cube in the start, and none of the volume of the water has been added. While in the end, in the 0.4%, they have added 2% of the original stock solution, and the volume of W is 8. So we can just work it out like this, that the zero point 0.8 will be 4.0, and this will be 6.0. This will then become, this will decrease, okay, the water will decrease as we move upwards. So this will then become 4.0, this will become 6.0, this will then become 8.0, and this will become 2.0. Even if this does not make sense to you, that how are we actually just you can say trying to figure out the water content and the volume of pee, what you could do is that you could just use the formula of the simple dilution, which I have already showed you, that it is m1v1 is equals to m2v2. So now you know that we are preparing a, a concentration of 1.6 from 2. So the original is 2.0. The volume we are going to be taking it out of it, we do not know that volume. So the which concentration are we preparing, it can depend on which you are going to be performing, like for 1.6, 1.2, 0.8. So uh, right now I'm going to be just placing 1.6 here, and the volume that we are actually preparing is 10 centimeter. Okay, so this comes out to be 16 over 2. So the V1 is actually 8 cm cube. It means we are going to be taking 8 cm cube of the original stock solution, and we are going to be adding 2 centimeter to make up the 10 centimeter cube of the 1.6% amylase solution. So this is how you work out the volume of E and the volume of W that has to be mixed, in case if you can't figure out directly. So now let's move on. During the investigation, you will be sampling at intervals and using the iodine solution. So now just recall at the moment in your mind, what is iodine solution used for? It is used for the starch presence. Okay, for the presence of the starch. The endpoint is reached when the iodine solution is a dark yellow brown. There may also be some specks of blue-black present in the iodine solution. These specks can be ignored. Okay, so carry out step 1 to 14. You have to prepare the concentration of the amylase solution as shown in the table 1.2 in the beakers provided. Okay, so you have to prepare these five concentrations. One is of the 2%, the other is 1.6, then 1.2, then 0.8, then 0.4. First you have to be preparing in these in the beaker. Most of the students, the mistake they do is that they just prepare it in the test tube in which they are going to be performing the experiment. You do not have to do this. Just prepare it first in the beaker, and then you are going to be taking out of it the required amount. Label the test tubes with the concentration of the amylase solution. So we are going to be then labeling the five test tubes with the concentration, like for example 2, 1.6, 1.2, 0.8, 0.4. Then we are going to be labeling the white tile with the numbers shown. Okay, they have already showed you a diagram that how you are going to be doing the labeling of the white tile. So put one drop of the iodine on the white tile at each of the sampling time. For example, they have just labeled the time. Actually the 15 shows the 15 seconds, the 30 shows 30 seconds, 45, 60, 75, 90, 105, 120, 135, 150, 165, and the last one 180. So we are going to be placing a drop of iodine at each of the spot on the white tile after we label it. Then we are going to be putting 3 cm cube of S into each of the test tube labeled in the step 2. So put 1 centimeter cube of the 2% amylase solution E into a test tube labeled 2.0. percent, use a glass rod to mix it, and then start timing. So now what are we actually doing? Let's just see what actually S is and why did we added it to the test tube. So S is the starch solution and E was the amylase solution. Okay, so now, when we add we add the starch into the amylase solution, then we are actually we are starting the timer, and then what we will be doing, I'm just assuming that we are, as the time passes, like first the 15 second, the 30 second, the 45 second, we will be placing a drop of each at the required labels on the white tile to see if how much of the hydrolysis have actually occurred. So in this case we are actually performing the opposite operation. For example, we test for the starch solution, but here what are we doing is that we are waiting it for to disappear, waiting it for to be the starch to be hydrolyzed. Okay, so as the time progresses, it is going to the blue black color is going to be just you can say vanish. After 15 seconds, use a glass rod to transfer a drop of the mixture from test tube into a drop of iodine that is labeled 15 on the white tile. Clean the glass rod with a paper towel. Repeat step 8 to 9, which is just like you can say after each 15 seconds we are going to be placing a drop, and record in A3 the time when the endpoint is first observed. If the endpoint is not reached at 180 second, record as more than 180. Clean the white tile with a damp paper towel and then dry the white tile. Make sure all the numbers are still visible. Put one drop of iodine on the white tile at each sampling time and repeat 6 to step 13 using other concentration. Okay, so they have showed us how we are going to be doing with the 2%, and now we are going to be performing it with the 1.6, the 1.2, the 0.8, 0.4. So this is going to be a lengthy experiment. So now what I'm doing is that I'm not actually performing the experiment. So what I'll be doing is that I'll be just deducing that what each concentration will have the effect and how much time will each concentration have to hydrolyze. So you know that as the amylase, I'm just writing the statement so that there is no confusion. As amylase concentration will decrease, the hydrolysis will also decrease. Okay, so you agree with that because there will be less enzyme to hydrolyze the starch. So now let's make a table here. So I have made this table. Now let's plot the x and the y axis. In the first column we will say the concentration of E, and its unit is percentage, and then time taken for color change. I'm not writing the full form because there is less space here for color change, and then its unit will be seconds. Now we have 5 readings: 2, 1.6, 1.2, 0.8, 0.4. So now let's divide it into 4 equal sections. So here comes the fourth one, and the last is the fifth one. So this is, we'll be writing the concentration of E, which is the independent variable, in an ascending order. Okay, so it is zero point 0.4, then comes the 0.8, then comes the 1.2, then 1.6, then 2.0. So now we can say that hydrolysis will also decrease as the concentration is decreasing. So let me just assume that 0.4 will have more than 180, and let's assume that this will have about, just let's see the time that we are being provided. So we have till 180. So now let's just assume it for it at 165, and we'll be decreasing the time taken for the hydrolysis or the color change. So this will be Let's assume it as 150, and this will then become, let's just you can say 120, and then the 2% it will have the most, the least amount of the time, as the hydrolysis will occur the most quickest. So it will be about, you can say, 90 seconds. Now they are telling us state independent variable in this investigation. So you know that the independent variable is actually the variable that we are changing. Like it does not have effect on any of the thing, but we are changing that variable. So it will be the concentration of amylase solution. This will be the concentration of amylase solution as we are changing it. So that is actually the independent variable. And what will be the dependent variable? Dependent variable is which is being dependent on the first or the independent variable. So what is the concentration of the amylase solution affecting in the case? It is affecting the color change. So that will be a dependent variable. Now carry out step 15 to step 23. Put one drop of iodine on the white tile at each of the sampling time as shown in the figure 1.1. Label test tube with letter U. Put 3 cm of S into the test tube. Put 1 cm cube of the solution U into the same test tube. Use a glass rod to mix. Start timing. After 15 seconds, use the glass rod to transfer a drop from the mixture to the test tube into the drop of iodine that is labeled 15 on the white tile. So we are going to be performing the same type of the procedure that we did with the concentration, but this time we are going to be doing with the U. So what is actually the purpose of performing all this experiment? The purpose is that we identify the concentration of the unknown solution, okay, the unknown amylase solution. So we will be performing it with this. So now let's just assume that U gives us 135 seconds. It gives us the color change at 135 seconds. So we can see from our table that U will most probably occur between 150 and 120, as the time is between this, so 135. So we can just write any concentration between 1.2 to 1.6. So let's just assume 1.4%. Suggest one source of error in the procedure described in the step 10 of this investigation. So these type of the question is the called the plan investigation, but this is a very small type of, you can say, this has very less marks, but often it often comes as 4 to 5 marks of a question, and it actually requires considerable planning, which I have also addressed this question in the revision question, so in the revision video. So you can actually watch it and just see that how we are going to be applying the modifications and the errors to improve our experiment. So it's just one source of error. So the source of error in this experiment could be the it is difficult to reduce the endpoint. So difficult to see endpoint, or you can say when it occurs actually. The other point that can be is that the volume that is being transferred, like they have asked us to just drop one a drop of it of the solution to the iodine. So that is actually, you can say, it varies. Like it cannot be just perfectly add one drop of the solution. So we could use you can say a pipette for it. Okay. Suggest how you could make one improvement to reduce the source of error stated in the part 4. So what we could do is that we could use a colorimeter. So colorimeter to give numerical values of endpoint as the colorimeter gives non-subjective results. Which is, what is non-subjective? Non-subjective means that the eye is not the only source that is actually measuring, because what we see that it can be a bit, you can say, when there is very light concentration of the star that is being removed, so there can be a light brownish color, but we cannot see it properly with the eyes, because our eyes have a certain limit and we cannot actually you can say observe certain hues and colors. So the colorimeter actually gives you non-numerical values. They are actually calculated values, not being based on the vision or anything. Gives non-subjective values. The other improvement that can be is that use color standard, or you can say the color chart or the color standard. So you can actually see the when the color change occurs. Discover how you modify the procedure to investigate the effect of pH on the time taken for the amylase to hydrolyze the starch. Okay, so now we will be using one concentration that takes least amount of time to hydrolyze starch. So now most of you will be wondering, why are we using the least amount of time, uh, the the concentration that takes least amount of the time. So actually we what are we doing is that we are finding the optimum pH, the optimum concentration. So the optimum or the most concentration will be with the which has the least, which takes the least amount of time to hydrolyzes, or you can say which takes the least amount of time to show the color change. And then we will be perform the experiment with 5 different pH values. So we have chosen the optimum concentration, and then we are using 5 different pH values. Also provide the numerical values. You can say it can be 2, 4, 6, 8, and also 10. So this is the how you are going to be investigating the effect of the pH on the time taken for the amylase to hydrolyze. And then record time each pH takes to show color change. Now B part says the student investigated the effect of different substrate concentration on the activity of the enzyme in the presence of the inhibitor. The concentration of inhibitor was standardized. The results are shown in table 1.3. So now they are requiring us to actually make a graph, and this is the easiest part of, I think, of the question ones that usually come up. But one of the student was actually asking me to address some of the problems that are being associated with the graphs. So let me just address it first. So so the type of the graph that we encounter in these type of questions is the most commonly the line graph, and after that the bar graph. I have just, you can say, just rarely seen a histogram, because they mostly use the line graphs. Like in this scenario they are also requiring us to draw a line graph, but how do I know that we are going to be making a line graph or a bar graph? So line graph is used when both columns have numbers, or you can say numerical values. So let me show it to you. So now you can see the both of them have numerical values, while the bar graph is used when x-axis has categories or the words. For example, in one question that I was solving, it was about like the sucrose concentration in different fruits. So the x-axis actually contained the name of the fruits like the grapes, mangoes, and like that, while the y-axis it contains contain the numerical values. So that is when we know that we have to make a bar graph. This is the understanding that you should have, but also the examiner actually tells you that you have to whether you have to make a bar graph or a line graph. The x-axis will have non-numeric groups like plant species or conditions. It can be anything but words, not numerical values. Okay, the y-axis is it will have numbers. And when are we going to be using histogram? Use when the data is continuous and the group. For example, it can be about the height, or you can say the frequency. Okay, the x-axis will have the intervals and the range of the values, while the y-axis will have the frequency of how many time it occurs, or things like that. So how actually we draw a graph correctly? We use a sharp pencil and a ruler for neatness. Label both axes with the correct variables with unit from the table they have provided. Okay, choose a sensible scale that use at least half of the graph paper. So most of the students, what they do is like, just like we draw a graph in the physics or the chemistry, they just do the same with the bio. So you do not have to make the scales in like in the decimals. Okay, you have to choose some sensible scale like zero point 0.5. It can be zero point 0.5, it can be zero point 0.1, or it could be zero point 0.2, like that, but not like some weird scale like 0.00375 or things like that. Okay, use a sensible scale that is actually more, you can say, it is more convenient to divide between the small blocks. Give the graph a title based on the data, but this is not necessary. Okay, because the examiner actually providing you the graph, he knows what is it is about, so just skip this part. But you have to choose a sensible scale. You have to label the axis based on the units that are being provided in the table. The other thing that I have included is the line graph rules. Plot the points carefully, and whenever you are plotting the points, you are going to be using this small dot with a small circle, but the circle that is actually encompassing it, it should be a bit small. Okay, not a large circle, or you can just use a small cross like this. Join points with straight lines. Okay, do not use a curve. For example, if I have these points, this is one of the point, this is the other point, this is the third point, this is the fourth point. I'm using to be just joining it with the ruled lines. Okay, not with the not a curve. Most of students, what they do is that they just draw a curve like this. Curve is not allowed in the biology graphs. Another thing, do not extrapolate the graphs. Do not draw or extend the line beyond the last point. Okay, for example, if this was not within the origin, if this was not meeting the origin, so you do not have to extrapolate or just, you can say, force it to join the origin. Just leave it as it is, or you can say, just plot the numerical values they have told you in the table. So this was the other point. The bar graph rules are leave gaps between the bars. Most of the students cannot differentiate between the bar or the bar graph or the histogram. Histogram do not have any spaces, while the bar graphs have. All the bars must be of equal width. For example, if this is one of the bar and this is another bar, so they both should have, just you can say, they both should cover five boxes, five small boxes, or any, you can just you can say, you can choose any of the number of the boxes you want, but all of them should then you can say follow that number of the boxes. Label categories clearly on the x-axis under each of the bar graph. Okay, under each of the bar. The bar graph has categories on the x-axis, while the histogram has continuous data. The line graph has numerical values on both. Bar graphs have gaps, while the histogram don't have. Purpose is to compare categories in the bar graph, while in the histogram it is to show frequency distribution. I've just provided some of the examples that are going to help you in identifying the type of the graph you are going to be making. So the graph type is line graph for the time, temperature, pH, or you can see the concentrations graphs. While the bar graphs are drawn when, for example, you are comparing the height, or you can say the concentration of, you can say, different fruits, or it can be the amount of the amount of the land that was uncultivated, or you can say type of that, age groups, and the number of the students or the heights that are being you can say represented using an histogram. So now let's move on to this question. So now plot a graph of the data in the table 1.3 on the graph in the grid in the figure 1.2. Use a sharp pencil. So now we can see that over concentration of the substrate, the range lies from 0.0 to 1.0. Now we can divide this on x-axis based on the number of boxes that we are being provided. So you are being provided with 5 boxes. This is going to be the first one, this is going to be the second, the third, the fourth, the fifth. So you are going to be dividing 1 by 5 boxes. So it comes out to be 0.2, 0.4, 0.6, 0.8 and 1.0, and this is going to be 0.0. So this is how you are going to be choosing a sensible scale. Now the x-axis has the concentration of substrate in in mole per dm. So this is going to be concentration this is going to be concentration of substrate in mole per dm cube. So I have labeled this, while on the y-axis it is the rate of reaction, arbitrary units. So I am also going to be labeling this first, rate of so this is going to be the rate of reaction, arbitrary units. And now we can see the range that from which to which values the rate of reaction lies. So it lies from 0.007, 0.000 to 2.25. So we are going to be, it is a range of 2.25. And how many boxes are being provided with? This is going to be the first box, this is going to be the second, the third one, the fourth one, and this is going to be the fifth one. So now let's divide 2.25 divided by 5. So it comes out to be 0.45. Now I have told you that you are going to be choosing a sensible scale. So 0.45 is not a sensible scale. You can round it off to 0.5. This becomes 1.0, then 1.5, then 2.0, then 2.5. Okay, now what is the first reading? It is 0.00, and the both of them start from the 0. So in this case it is starting from the origin. So you are going to be plotting that point. This is the origin point. Now 0.2 and 0.750. This is 0.2, and now let's see where this 0.75 occurs. 0.75 occurs between the midway of the 1 and the 0.5. So this is going to be the second point. Now the other point is 0.4 and 1.375. Okay, so 1.5 plus 1 divided by 2, so it is 1.25. This point will be 1.25. So now the two small boxes above 1.25 will be, let me just check. Okay, two small boxes the above 1.25 will be 1.375. So this will be the point of 1.375 and 0.4. Then comes 0.6 and 1.8. So where does the 1.8 occur? We have a distance of 0.5 in one big box on the y-axis. So let's just check where does 1.8 occur. Another trick that I want to show you is that in case if you can't find out where does it occur, what you have to do is that you are going to be looking for the the value that is nearest to the value. For example, if I'm just finding out 1.8 right now, I'm going to be looking for the value that is the nearest, and you can see the lowest and the nearest value to it. So it is going to be 1.5. Okay, now what I'm going to be doing is 1.8 minus 1.5, and then I'm going to be divided by the least number, or you can say the least representation of one box. For example, this one small box represent 0.005, 0.05 actually. So it then divided by 0.05, so this will come out as, divided by 0.05, it will come as 6 small boxes. So I know that 1.8 will be here. 0.8 and 2.175. Now let's do the same for it. 2.175 minus 2 divided by 0.05, it will come as 3.5. So this will be about 3.5 units above 2.0. Now the last one is 2.25 and 1.0. So I know it will occur between, just between mid of the 2 and 2.5. So this will be the last point here. Now I'm going to be joining these all points with straight line. Okay, one straight line from here to here, and other from here to here. This is going to be the second straight line. This is going to be the third straight line, the fourth one, and the fifth one. So I hope it's clear. Let's move on. Use your graph in figure 1.2 to determine the rate of reaction when the concentration of the substrate is 0.4 mole per dm cube. So 0.45 actually. So this is 0.5, and 0.45 will be in between the mid of this. So now you are going to be showing it with dotted lines like this, but using a ruled line. Okay, not like not like I am doing. You're going to be use a ruler to draw a dotted line here, and when it reaches the curve, you are going to be tracing the concentration, the rate of reaction at which this concentration occurs. So it is 1.45. Now the Vmax of this enzyme is 2.25 arbitrary units. Using information of the graph, state whether the inhibitor is competitive inhibitor or non-competitive inhibitor. So now let's check. It is so as we can see that over Vmax is actually also reaching the maximum rate of reaction, is actually reaching to 2.25. We would say it is a competitive inhibitor. Why? Because the Vmax still reaches that point, and increasing the substrate concentration actually reduces the effect of inhibition. So this is the knowledge which we will be applying from what we know about the enzymes. So 2.25, so it will be competitive inhibitor. Now we will explain it as the Vmax still reaches 2.25 arbitrary, hence the inhibitor competes with the active site. Let me just remove this. Competes with active site and binds with it. The effect of inhibitor can be reduced by increasing the substrate concentration. So you are not going to be writing about anything about the km value, as we do not have some graphs to compare. Okay, they have just given us the Vmax of the actual enzyme. So this was the answer for this. Let's move on to question number 2. So M1 is a slide of stain transfer section through a plant leaf. A1 says draw a large plant diagram of the region of the leaf on M1 indicated by the shaded area in figure 2.1. Use a sharp pencil, use one ruled label line and define the palisade tissue. Okay, palisade tissue, not the palisade. You can say not. You can just use a line to identify the palisade mesophyll tissue. So this is the slide which I have chosen. Actually, I am not using the actual slide which was actually provided in the examinations, but I'm just you can say it is also a transfer session to a plant leaf. So this will be the diagram. This will be the slide from which I'm going to be making the plant diagram. So I have already made this plant diagram from it. Let me just drag it here. So this is going to be the plant diagram. So actually it is going behind the page, so let it just remain at the side of it. So you can see here, let me just zoom out a bit so that we can make a comparison between it. So you can see here that I have at first draw the structure of it, like this is going to be the upper epidermis. Let me change the pen so you can see it clearly. So you can see that this is the upper epidermis. First of all, I have made this with a double line, and you could also see a third line at the topmost. It is actually a waxy cuticle. This is the waxy cuticle. The second one is the epidermis. Then this is the vascular bundle. This is a small vascular bundle actually, with this part being the xylem. This is the xylem, and this is the phloem, and this is the you can see the cambium between it. Now this is the large vascular bundle in the between, which you could see. This is the large vascular bundle. You are going to be showing the proportion of it, that how it is being divided. The xylem covers the more than half of the area. Then this is the xylem parenchyma. This is then this is the phloem parenchyma. So I actually told you that how that the xylem actually covers the more than half of the area. Then this is going to be the palisade mesophyll cell, which you can see as longitudinal columnar cells here, and uh, from here the purple ones. Okay, this is going to be the palisade mesophyll cells. Then this is going to be the spongy mesophyll cells. As you can see, they are just in you can see unorderly arranged, and in they have intercellular spaces as well. Then you are going to be showing a stomata as well. You can see this small gap. This is here, you can see. So you are going to be looking for the stomata, and you are going to be showing it. Then comes the lower epidermis. Also look for some slides a bit carefully, because some of the plant leaves also contain cuticle on the lower epidermis. So this was the plan diagram of it, and we are going to be labeling this as the palisade tissue. Okay, this is the palisade tissue. Now let's move on. Observe the cells in the upper epidermis. As I told you that they mostly ask about the epidermis, the cortex, the pith, the xylem cells. So I've already addressed this issue on in the higher magnification drawings video. So select a line of 4 adjacent epidermal cells. Each cell must touch at least one other cell and make a large drawing of line of 4 epidermal cells. Use one rule label line to identify the cell wall of the one epidermal cell. So I have drawn these epidermal cells from that slide. We are going to be using our own knowledge to draw the vacuole. The nucleus is visible actually, but even if it is not visible, you are going to be making the nucleus. So this is going to be the cell wall. Let's move on. Figure 2.2 is a photomicrograph of a strain transfer section of a different leaf from M1. You are not expected to be familiar with this specimen. Identify three observable differences other than the color between the leaf section in the 2.2 and the leaf section in M1 slide. Record and these three observable features. So let me just discuss it first. You can see these structures, the protruding structures. These are the trichomes. They were not present in that dicot leaf. So first one is the trichomes. This is one of the observable features. Now, what can be the other feature? Just let me make it a box. Okay, what can be the other observable feature? You can see the palisade mesophyll and the spongy mesophyll. They occupy less space here. You can compare this gap with the slide gap. So you can also write about the area covered by mesophyll tissue. The other thing that you can write is that it contains one vascular bundle and it is also smaller. You can also write about the size. So you can write about the number of vascular bundle. So this is another difference that you can actually write about. So I've already discussed the the observable difference. Hope you can write it in the paper. So now let's move on. Figure 2.3 show a diagram of a stage micrometer scale that is being used to calibrate an eyepiece reticle. The lower one is the eyepiece reticle. This is the eyepiece reticle. The length of one division of this stage micrometer is 1 millimeter. So this is the stage micrometer. Let me tell you one more thing, that most of the people are actually confused in one division. One division is a distance between two lines like this, like these two lines, okay? So this is actually called one division. Okay, this is called one division. So this is actually 1 millimeter. Now let's find out the distance of one eyepiece reticle unit before they ask us about the actual length. So we know that from 50 to 90 is actually equal to 1 millimeter. So 50 to 90 is actually equal to 40 eyepiece units. I'll be using EU for short form. It is actually equal to 1 millimeter. So 1 EU will be equal to x millimeter. You can just cross multiply to get the answer. So it will be 1 divided by 40. So 0.025. Okay, 0.025 millimeter. Now using figure 2.3 to calculate the actual length of one eyepiece reticle unit. So I've already done that. Let's just drag it down to this area. Now they've also told us to show a working and give your answer in micrometers. Okay, we're going to providing the answer in micrometer. So it is 0.025 millimeter. Multiply it with 1000 as we are moving toward the smaller unit. So it will become 0.025 into 1000. It will become 25 micrometer. As it is the suitable micrometer is the suitable unit for the plant structures. Figure 2.4 is the same photomicrograph as shown in the figure 2.2. Data was taken using the same microscope and the eyepiece reticle. The eyepiece reticle scale has been placed across the midrib of the leaf section. Use the calibration of the eyepiece reticle unit from the C1 to calculate the actual thickness of the midrib of the leaf section. Show your working and use appropriate units. So we can see that the midrib lies from the 15th mark to the 60th mark. Okay, so the eyepiece, the midrib length is 60 minus 25, which will come as 35 eyepiece units. So let's multiply it with 1 unit, which was 25 micrometer. Okay, so 35 into 25 comes out to be 875 micrometer. So we have a perfect answer, 875 micrometer. So just a possible function for the structure labeled A. So this is the structure labeled A, which I told you is a trichome for sure. So what is the function of trichome? They reduce the surface area to decrease rate of transpiration. So this will be the 1 mark answer for this part. The paper is finished, and this was what I was actually expecting for the paper 34 of 2025. So this was the paper, and we have solved the full paper. One question from the enzymes and one from the microscopy section. So subscribe and share this video with anyone you think it will help. I'll be meeting you in another video. Till then, good luck for the paper and goodbye.